Level 6 Diploma in OH&S, Toxicology, the study of the negative effects of chemicals on living things. Chemical is considered toxic depending on how much of it is necessary to cause harm, or how easily it can enter the body. LD50 and LC50. These terms derive from laboratory tests on animals. LD50 is the dose which, when swallowed, injected, or applied directly, kills half the test subjects. This is defined as the dose required to kill half the members of a specific animal population when entering the animal's body by a particular route. LD50 is a general indicator of a substance's toxicity within a short space of time. It is a measure of acute toxicity. LC50 is the concentration of a chemical in a test atmosphere that kills half the test subjects within 1 hour when inhaled. LC stands for lethal concentration. LC values usually refer to the concentration of a chemical in air, but in environmental studies it can also mean the concentration of a chemical in water. Why LD50 and LC50 are values a measure of acute toxicity. Acute toxicity is the ability of a chemical to cause ill effects relatively soon after one oral administration or a 4-hour exposure to a chemical in air. Relatively soon is usually defined as a period of minutes, hours, up to 24, or days, up to about 2 weeks, but rarely longer. How are LD-LC50 tests done? In nearly all cases, LD50 tests are performed using a pure form of the chemical. Mixtures are rarely studied. The chemical may be given to the animals by mouth, oral, by applying on the skin, dermal, by injection at sites such as the blood veins, muscles, or into the abdominal cavity. The LD50 value obtained at the end of the experiment is identified as the LD50, oral, LD50, skin, LD50, etc. As appropriate, researchers can do the test with any animal species, but they use rats or mice. Most other species include dogs, hamsters, cats, guinea pigs, rabbits, and monkeys. In each case, the LD50 value is expressed as the weight of chemical administered per kilogram body weight of the animal, and it states the test animal used and route of exposure or administration. Example: LD50, oral, rat, 5 mg per kilogram, LD50, skin, rabbit, 5 kg. So, the example LD50, oral, rat, 5 mg per kilogram means that 5 mg of that chemical for every 1 kg body weight of the rat, when administered in one dose by mouth, causes the death of 50% of the test group. If the lethal effects from breathing a compound are to be tested, the chemical, usually a gas or vapor, is first mixed in a known concentration in a special air chamber where the test animals will be placed. This concentration is usually quoted as parts per million, ppm, or milligrams per cubic meter, milligram m3. In these experiments, the concentration that kills 50% of the animals is called an LC50, lethal concentration 50, rather than an LD50. When an LC50 value is reported, it should also state the kind of test animal studied and the duration of the exposure. Example: LC50, rat, 1000 ppm 4 hours, or LC50, mouse, 5 mg m3 2 hours. How should an LD50 value be used? The LD50 can be used as an aid in developing emergency procedures in case of a major spill or accident, to help develop guidelines for the use of appropriate safety clothing and equipment. For example, if the dermal LD50 value for a chemical is rated as extremely toxic, it is important to protect the skin with clothing, gloves, etc., made of the right chemical-resistant material before handling. Alternatively, if a chemical has an inhalation LC50 value which indicates that it is relatively harmless, respiratory protective equipment may not be necessary, as long as the oxygen concentration in the air is in the normal range around 80. For the development of transportation regulations, as an aid in establishing occupational exposure limits, as a part of the information in safety data sheets, remember, the LD50 is only a ballpark figure so that lethal toxicity can be compared. It says nothing about levels at which other acute toxic, but non-lethal, effects might occur. The LD50 is only one source of toxicity information. For a more thorough picture of the immediate or acute toxicity of a chemical, additional information should be considered, such as the lowest dose that causes a toxic effect, TDLO, the rate of recovery from a toxic effect, and the possibility that exposure to some mixtures may result in increasing the toxic effect of an individual chemical.